most of the time I'm doing medicine, Department of Medicine, working as a clinician, and my subspecialty is nephrology. So I am a nephrologist working with chronic and acute kidney patients. In chronic kidney disease, the life expectancy is just uncomparable. Our patients have 3 order magnitude of lower life expectancy as compared to the general population. It means that at age of 25 is similar to that of 78. So it's huge difference there. And the most important pathology behind that is the calcification, the mineralization. And this is one of our patients developing medial calcification. It's a diffuse. The density is close to the bone, and at age of 38. So it's young, an older lady. One of my patients, this is the aorta, and this is the vertebra. So the medial calcification is a huge afterload of the myocardium, therefore dilated cardiomyopathy develops. And the intimal calcification is responsible mostly for the ischemic diseases, stroke, heart attack, peripheral artery disease. And this is the intima. And very close to that, there is a focus of mineralization. And this is one of our patients' specimens of carotid artery removed by the surgeons. The aortic stenosis and insufficiency is due to the calcific aortic valve disease. Heart surgeon removed this in our university, and we got the specimens from him. And the calciphylaxis is a small vessel calcification. You can see the small vessels and the obliteration of the vessel provoking an ischemia, and because of the ischemia, there is necrosis, and the mortality is almost 100%. So, being a nephrologist, our research is one of our focuses is on calcification, remodeling of the vasculature, and doing that, I brought a small movie for you. We isolate cells and working on the tissues, human tissues, and isolating the cells. This is one of our patients' carotid artery specimens, smooth muscle cells, and many years ago. And they should move around and synthesize extracellular matrix. But here, you can see that it's a grayish dots, the extracellular dots is over there, and it's getting bigger and bigger, and vesicle formation is there. So I was fascinated by this movie, and have been working on this. This is the scanning electron microscopy. It's like a spider net. It's a close-up ball, and we performed adducts, identifying the various type of minerals. So it's hydroxyapatite. So it's bone. It's not calcium phosphate precipitate. So these cells are supposed to be smooth muscle cells, but they are not. They are osteoblast-like cells. And similarly to one of our patients' aorta, dying of dissection, so these smooth muscle cells are making bone in the tissue. So the calcification is a bone formation in vascular biology in these disease states. And this is one confirmation of that. This is a calcific aortic valve, and the RUNX2 is a transcription factor, driving the osteoblast activity. And as you can see, there is a translocation of the nucleus in these cells. indicating that the reprogramming of the cells are occurring in human tissue. That's the same for the carotid artery and the aorta. So we have been working on this area for 30 years and realized that the calcification, the mineralization is associated with hemoglobin deposition. And analyzing the hemoglobin, we identified sulfhemoglobin. So there should be free hydrogen sulfide gas within this tissue. And in the past years, I got a few patients of sulfhemoglobinemia, and the pathophysiology is very interesting. So the hydrogen sulfide gas, it's a poisonous one. Bad egg, it smells bad. And so at the beginning, I thought that it's a pro-pathogenic factor. But it's turned out the opposite. After many, many years, this is an in vivo experiment, ApoE knockout developing aortic calcification, and exogenous hydrogen sulfide prevents the calcification, and in these cells there is no Renix activity. And so we have hydrogen sulfide generating system. It's a system which is making gas within the tissue, within the aorta, the huge arteries. And even within the valves, aortic valves. And this is the pathogenesis of the transition of these cells to osteoblast-like cells. And in the past 20 years, we identified many controlling mechanisms within the smooth muscle cell, valvular interstitial cells, which can control the transformation of the cells into the osteoblast-like cells. Later, we just realized that this is not so simple because we have been detecting the expression of the machinery. We realized that the genes are even upregulated. So calcification, upregulation, where we had high expression of H2S generating system, we had high calcification, high inflammation. And didn't understand that. That's what is true for the aorta and other tissue, and even in calciphylaxis. So we went on and performed, actually improved ourselves when we are able to measure bioavailable H2S and realize it's down. So the gene is up, the gas is down. So it's funny, so we thought that it's a dichotomy and the enzyme is not working. But performing metabolomics, that's not true. We realized that our thinking was wrong and realized that the other product of the system, the lanthionine, is increased. So it was true all over the tissues, what we identified. And the question is that we have a gaseous molecule, exogenous one. We don't have treatment in our patients, for our patients. So no, there is no specific treatment for calcification. Even in diabetes or aging, when we're aging, the calcification mineralization is there. And we had approach and targets mechanism to control that. But we didn't understand how can we control those. So we knew that the gas concentration is down, the machinery is up. So there should be some kind of consumption. So we turn to mitochondria. This is the terminal oxidation that we don't have too much time, but I guess you are familiar with that. And there is a hydrogen sulfide oxidation within the mitochondria, and consisting of 5 enzymes. And we have been going after the machinery within the tissue, human tissue, and realized that mitochondria consumes enormous amount of H2S. So the mitochondria deplete the H2S in the tissue. We don't know why, though. We have been working on that. So the therapy. So we confirmed in consortium over disease involving the States and one of my friends over there in Britain. And we have been working on making substrate. And realized that we should dock our donors within the mitochondria. This is tissue from mice. This is the bone. This is the inflamed cells. These are valvular interstitial cells. And exogenous donor docking to the mitochondria, preventing the disease in animal. Stage 5. The preclinical stuff is done, the clinical phase we have been working on. So in medicine, we propose that even in diabetes or other triggers, there are calcification ones and pro-inflammatory ones. And early on, we realized that all of the triggers increases the machinery. I label just one, which brings up the level of H2S. It's depending, sometimes it takes months or days. And after a while, this is an adaptation period. A prolonged period alters the mitochondrial function. Mostly, one of those is the H2S oxidation, bringing down the CSE and allowing a pathophysiological remodeling, including calcification. So this is a 25 years of work or so, coming from a basic research experiments, followed by exploring the pathogenesis. And you can see that we had hypotheses, and it turned out the opposite, not just one time, but many times. And if we want to prove our hypothesis, we would fail. Therefore, we had to accept our data. And finally, we got into pharmacokinetical experiments and find a molecule which can be a medical approach as well. So this is our laboratory. Was established in '94. One of them became the president of American Society of Nephrology, Zarju here. And so one part of our research is focused on, but of course we have many others and dedicated. These guys they are good, Nature Medicine and JCI, so they are good. This is our university of Debrecen, and you are welcome over there. Thank you.
Thank you very much. We have time for one quick question. If you have anything to ask about the calcification and the impact of SH2, just don't be shy, come forward, and we can do that later on. Now I would like to hand over the recognition.